Well, by word of brief introduction, I want to say a little vignette about Riccardo, which is so Riccardo comes from the University of Chicago, and I would say like for those of us who have been in the biophysics sort of culture for a little bit, there's this like maybe one of the few success stories we can reasonably grasp for is kinetic proofreading. This metaphor that like one of the ways that biology achieves really wonderful fidelity is by spending energy. Um, and like maybe it's one of the only vignettes that kind of resembled the sort of Dirac out of Blackboard deriving spin 1/2, but for biology. It's like from a Blackboard calculation, you can calculate like the existence of mechanisms like this. But that being said, like if you actually look at like what people are able to conclude about the natural world from this metaphor that's supposedly so successful, it's hard, it's messy, it's like people argue about it all the time. And I think that Ricardo is one of the clearest speakers on this and thinkers on this topic.
Mm. Well, thank you, Gabe. Oh, thank you for having me here. So the title of the talk of today was Behind the Scenes of My Evolution of Error Correction Mechanism. And behind the scenes because I want to give a sort of like the idea of how the project started, which is very different from what the project became. And I'm also going to talk through a bit what was the communication between theory and experiments that made all this project possible also. And to start, I want to tell you what the project is. So just in a couple of words, what's the outcome of this project? And so the way I motivate it now is that, great. You have a cell, and in order for, a cell to survive, there's a lot of things that need to happen correctly inside the cell. There's going to be, in the nucleus, there is DNA, right? And DNA needs to be copied. It needs to be copied with a certain accuracy for genetic information to be transmitted across generations. So we have DNA replication. And there's a certain error rate that I can call mu. And this error rate needs to be, let's say, low enough for permitting this cell to, to be alive in the future, to keep being alive. But, there's many other things that happen in the cell. For example, there is ribosome. Let's say that this is our ribosome. And the ribosome itself needs to be it actually goes a process of self-assembly. So it needs to be assembled in its final structure. And this process also, well, you need to have a given ribosome in order for it to function. So the self-assembly process also needs to be accurate. And, there's very many other examples. I just give another one, which is like, the formation of like protein chains. So just, amino acid chains. And that also is a process where you need to put the right amino acid in the right place to form a protein. And this is what, I call as ordered process that makes the cell alive and be able to keep going. And all of these processes are actually working in this precise manner thanks to error correction. So error correction happens in all of these. And in the case of DNA replication, you might have the insertion of a wrong base. So let's say that here I'm replicating the strand, and I put a sort of like a wrong nucleotide here. But what happens is that this wrong nucleotide can be removed and the process can restart again, where you will have then the right nucleotide. So this is what is error correction, is just removal and a sort of like backward process that is bringing you back to the beginning and be able to start again and insert the correct nucleotide. And in similar ways, in the self-assembly of the ribosome and in protein chains, you have these steps that are sort of like bringing you back to an original step and giving you a chance to restart. So I see error correction here just as a sort of like backward-facing process. And our project is sort of like asking, okay, we do have this error correction that happens in cell and it's necessary for them to function, but what is sort of like the evolutionary origin of it? If you think about the biophysical or even biochemical pressures that are there at the origin of life, or that make this error correction possible. And our project is sort of like giving a possible answer, at least to some parts of this question, with some caveats. And what we found out is that actually these mistakes that happen cause a pausing in the process, meaning that here, for example, I introduce a mistake, but if I introduce a mistake, the replication will actually continue at a slower pace. Means that a mistake will slow you down. And this is true also in other cases. In the case of the ribosome, there are actually sort of like checkpoints that are going to make you slower. So they are going to sort of like pause. And in similar ways in the formation of amino acid chains. And what we find out, as Gabe was saying, is that usually when people think about this process, you would say that the faster you do something, the more mistakes you're going to put in this process. But actually, we sort of like flip this view by finding that the faster you are, the better you are, because these mistakes are actually, if you do a lot of mistakes, you're going to be slowed down. So actually, this is sort of like something that I will come back later when I talk about theory and experiments. But this is sort of like saying that, if mistakes slow you down, what we find is that, faster is actually more accurate. And then why does this help us understanding how error correction can evolve? And the hypothesis is that, if you are faster and more accurate at the same time, then you can actually think that if you're a system that is under selection just for the speed of completion of any of those processes, so DNA replication, self-assembly or the formation of the protein chain, then you're going to have a pressure to also be more accurate. So as a byproduct, you're going to evolve error correction. The idea is that error correction is going to remove these mistakes that slow you down. So you just evolve error correction as a byproduct of like trying to be fast. So now, this is just to frame so the idea of our project and our main result. And then I think what's interesting in the context of this workshop is like telling you how the project started, because I joined so I'm a postdoc at UChicago with Arvin Morgan, and I joined in July 2021, so a while ago. And I've been working on these topics and related offshoots for all this time. So, but when I joined, I was not thinking about any of this particular framing, right? And I think it's interesting to see how this came to be. And this came to be because of some experimental work that a colleague was doing in the lab, where we were having different DNA polymerases. So DNA polymerases, they copy DNA, so they are responsible for, replicating DNA with a certain accuracy. They introduce errors at a certain rate. And these DNA polymerases that we would add in the lab, they had different error rates. So there will be this one, which would be like, let's say, very accurate. There was one, let's say, medium, and one that was like very error prone. And so I'm not going into the details, but we have a platform that we can use in the lab to put these DNA polymerase into yeast. And once they are into yeast, we can then sort of like look at how fast they are at replicating, and also looking at how error-prone they are. And, we were working with these DNA polymerases, not because, we were interested in any of these, but actually because we wanted to compare the rate of, if you're a more mutagenic polymerase, you're going to introduce more errors in the process. And we wanted to study whether that could be actually advantageous in some sort of like regime of evolution. If you introduce more errors, you might think that, that could be helpful in some stressful conditions. But actually what we find out is that, well, by measuring these two traits of like speed and error rate, what we find out is that the very accurate polymerase was actually the fastest ones, and they were sort of like, if we rank them by speed, you would have, the green one to be the fastest one, and then the medium one, and then, sort of like the more error-prone one to be the slowest. So we were already puzzled by this result. And that's when I started my postdoc, and I said, okay, we need to understand why this is true from a theoretical standpoint. And I said, okay. So at first it was DNA polymerases. So, you go back to what are the models of DNA replications that are out there. And as Gabe said, there's like a very large literature of these models that started as sort of like in the 70s. So there's like 74, 75 papers by Opfield and Independent Nino. The sort of like, we're asking the question of like, DNA replication as an equilibrium process that doesn't consume energy cannot explain the accuracy that is seen in biology. And what they came up with is that a way that is what is called kinetic proofreading of sort of like achieve accuracy by coupling to some sort of like energy driving that in biology, it's like NTP hydrolysis that happens in the system. But, if you take these standard models from many years ago and you look at what trade-off you would get between the speed and accuracy, you actually find the opposite one. You find that, as I said before, what you expect intuitively is that faster is more error-prone. So this is where I came in. And I sort of like, the project at the beginning was like really, how these two are related. So why, how, what, and the question in the end was like, what is missing the models of like, these DNA replication that are actually could give us possibly this opposite result. And at first I went in a sort of like direction that was interesting, but didn't turn out to be so interesting. And then we were talking with a newly appointed professor at Chicago, Jack Shostak, who now works a lot on origin of life. And, we were talking about, actually, just abstract, because, you read all these kinetic proofreading paper and they all make sense, but it's just, they did an effort of like trying to connect to experimental data, but sometimes, you sort of like lack the real description of what a polymerase does. And you sort of forget and you think that the polymerase is going to, be exactly the model that is in your theory paper. But like talking with Jack, we sort of like went back to like the basic of like how the DNA replication actually works from an experimental standpoint. And so Jack made us notice the fact that what I told you before, that if you are, let's say that, how does it work? That if I have my strand of DNA that needs to be copied, let's say AT-ACG, I can have a good copy up to there, but I could also have the same polymerase making an error here. A. And Jack told us, oh, well, these would be two very different scenarios, because if you have an error, then your nucleotide here, there is this 3' hydroxide part that the nucleotide would be misaligned, and the DNA polymerase will be much slower at incorporating this base. So this is what I was telling you before. So this is where we were like, oh, okay, this makes sense. There's like hundreds of experiments that actually look at the extension rate past a mismatch in DNA replication. And this was never introduced in the 40 years of models that were actually looking at DNA replication and kinetic proofreading. So now, what I did was the obvious thing to do, which was just taking the off-field kinetic proofreading models and just add this feature of this slowdown that is called stalling. Because, well, pretty obviously you're stalled after this direction of mismatch. And then ask the question of like, okay, is this what was missing? So if you introduce this ingredient, can we actually invert this trade-off and see that more accurate is faster? And I mean, intuitively, you can see that it's not so surprising, because if you start to introducing an ingredient that is telling you that mistakes are costly in time, then of course, if you are faster, you are more accurate in a way, right? So that's what happens. So if you start introducing these terms in the models, you would get what we have found experimentally. And then that sort of like got us excited, because you're like, okay, we have these few points in our experiments that are telling us this. We have a theory that is sort of like, adding an extra feature that exists experimentally to this model, that is saying that this, let's say, is not really true anymore, but the opposite is true, that more accurate is faster. And then we got excited because of the hypothesis that I told you about. And the hypothesis was that selection for speed of completion can evolve error correction as a byproduct. And now, I mean, is that obvious to you? That I feel, if you
forbidden word, Riccardo.
Well, no, I'm saying I don't. No, I know obvious is never. It's not obvious to me either. But what I'm saying is that I feel, because someone actually has told me, oh, why do you even? I mean, once some people have objected to me, oh, once you tell me that more accurate is faster, then of course, like if something that is fast is more accurate, then if you just select for speed, you're going to get things that are more accurate. But I was not. I didn't think that it was obvious. So, Aditya, you had a question?
Yeah. So it seems like here you had to assume stalling to see this result, right?
Yeah. Stalling.
One question I might ask is like, could you argue that stalling is actually selected for by evolution? Because if you have stalling, then you select for more fidelity, which then allows your organism to get more.
So this is a good question. It's a question that we haven't really touched yet. So we don't know whether stalling. We don't know whether stalling was selected for that or not. This stalling property seems to be intrinsic to the nature of the nucleotide. So how the genetic code is, like how the genetic code is.
Yeah, and stalling a physical factor, so it's a feature of biology that came from sort of evolutionary pressure.
Yeah, so that's debated, but I think it's some sort of like an open question to dig into what are the evolutionary pressure to the evolution of the stalling. Yeah. But yeah, so as I said, I found that introducing the stalling gave us the more accurate faster trade-off, then we got excited because we were like, okay, then we could actually start answering this sort of like question of like, we just have these systems and we just select for the speed of them, and the speed is a sort of like basic fitness to us for like, because you are a cell or you're some sort of like, DNA replication, you just select it for the speed. And well, if you're a cell, it's a sort of like tautological fitness in a way, because a cell wants to reproduce fast, right? And then just selecting for speed, I wanted to show whether in a simple model, I could actually evolve error correction. So that's what I did. I went back to this modified upfield model with the stalling, and I sort of like, looked, okay, can I just select for speed and see whether I get that I magically evolve these kinetic proofreading mechanisms? And yes, so I sort of like have an in silico check that it works. And then something else that we did was, again, say, okay, this is cool, but what's the experimental sort of like ground that we have to justify actually this theory? So we had these 3 points, and this setup that as the DNA polymerase in disease system was developed by another lab that published, a paper where they were doing whatever they wanted to do. And I sort of realized that in the SI of this paper, there was a table with 150 data points where they had an Excel table, so never plotted, where they actually measured these 2 traits for 150 different DNA polymerases. And these DNA polymerases had like a range of error rate and a range of speed. So also there, we sort of like said, oh, yeah.
Are they all from yeast? Or are you talking about 150 kinds of polymerase within a single species?
Yeah, this is a good question. So these polymerases are viral polymerases that are part of a toxin-antitoxin system that exists in the cytoplasm of diseased cells. So they're not DNA polymerases that replicate the genome of yeast. They're just a sort of like defense mechanism that somehow was acquired by diseased strain and used. Yeah.
Do you know anything about the natural history of them? Because it seems like if you could understand why they all coexist and are natural, are you getting there?
Maybe. I mean, the natural history, you mean what the natural history, thinking about sort of like what is the sites that are like looking at the natural history, meaning what is more conserved in them, or
they all coexist. Maybe they all fill a unique niche, or I want to use the word purpose very carefully.
Okay.
Like
no.
Good reasons to have fast and good reasons to have slow.
Yes. No, actually, it's good to be that you asked this question, because so these variants are all like made in the lab. So there is a wild type polymerase that is what exists in nature. So nature is just a sort of like wild type polymerase that is actually very accurate and also very fast. And then all these variants are just built in the lab and then studied. But wait, you also asked another question. But yeah, maybe it will come back to mind.
Ability.
Yeah.
Even.
Yeah. So natural, but yes. Oh, but yeah, what I wanted to say is that, so actually in this case, so I told you that this comes from like a toxin-antitoxin system. And the toxin-antitoxin system is good if it is copied at a very fast rate, because you want to have a lot of copies of whatever that, defense mechanism is. So in that context, actually thinking about a selection for speed from an evolutionary standpoint actually makes sense, because this DNA polymerase had probably gone some sort of like pressure to be fast, because you want, because of like copying this defense mechanism a lot, so that it could have a lot of it. So like this is important in yeah. Mm-hmm.
Relating to the variation, for the induced variations, were these randomly induced variations? Do you know?
Yeah, I know, because I'm also doing it in the lab also, which is where I'm going to. But so originally, these variations were induced because the authors of these, let's say, the data from the SI of this paper, and this group is a group led by Chang Liu at UC Irvine. So they wanted to find very error-prone DNA polymerases, because they wanted a very error-prone DNA polymerase to sort of like speed up the rate of evolution of this piece of DNA. So they were sort of like targeting more error prone. They were selecting for, yeah. And yeah, but so the question is like, okay, we also, this is like another moment where there was a lot of excitement because we sort of like found a gold mine of data that was not plotted. And it's actually the data where, these two traits are sort of like, the traits that would tell us a bit more if this trade-off of like more accurate is faster holds. So then, of course, plot the data, and then you have error rate, and then, well, here I'm writing the inverse of speed, which is the time to replicate. And what we find is that actually, the ones, so on this axis would be like low error rate, low error, and here it's high error. And when we plot this data, what we find is that the low error ones are actually, down here. And then these 100, around 100, actually it's more around 200, right? All these different points in a part of this trade-off plot between the time to replicate and the error rate that has a clear front. And this front is the front that is telling us that if you are accurate, actually you're going to be faster. So it seems that because if I sit on this front, here I have low error and it takes the most little time to replicate, and here I have a high error and it takes a lot of time to replicate. So it's okay, we plotted this and we said we got even more excited because it seems that that our hypothesis was sort of like supported by this large data set. And this is also a quite large data set for any DNA polymerase. So this is a particular DNA polymerase, but there are not so many data sets of polymerases where you have these two quantities measured for such a high number. So, okay, that's good. And then, before I mentioned the current framing of the project that had DNA replication and all the other things on the cell. And this is because once we saw these, we were sort of like convinced that there could be something true, but we also started thinking of, okay, how general this mechanism is, like how general it is for processes to sort of like have these slowdowns and also be selected for speed, and this selection for speed evolving these error correction mechanisms. And so we started thinking about which other processes other than DNA replication could fall into this category. And one that we started working on was self-assembly. And this is, again, just theory work. And the idea is very simple. Once you do it for DNA replication, in the sense that you just need a model of a self-assembly structure that could just be sort of like little Lego tiles, and then need to come together, and they need to form a particular structure. And again, here, if you put the wrong Lego tile there, it will be very, it will have a wrong shape, so like sort of like off-target interactions. It will be harder to continue the structure so that you're sort of like slow down. And in physics term, it's what happens is that there's a kinetic trap. So the system is sort of like trapped there kinetically, and it takes a long time for it to proceed. So, this is sort of like the stalling equivalent. So showing, in the end, what we sort of like realize is that many systems have this stalling property, that given a mistake, you're going to actually be slowed down. Yes, go ahead.
Just to clarify, when you say self-assembly, are you talking specifically about like protein folding, or are you talking about more general, like different structures?
Yeah, so for self-assembly, in the model that we considered is literally just a set of tiles, so it's very abstract.
Okay.
And I think for protein, I mean, it could be sort of like pushed into proteins, but it's not what we think about. It's more literally self-assembly of like a tile set, and then you could put it like back to, for example, the ribosome, or yeah, viral capsid is another example where it is also known that they need to be assembled in a self-assembly in a certain way, where there's also stalling. Protein folding is very related, but I think it's you know, there are chaperones proteins that help proteins fold. So I think the description doesn't really map to the self-assembly one that we had in our theoretical work.
Yeah, I mean, there are still energy landscapes, and
Yeah.
proteins can land in different valleys and get stuck there. Even, you know, chaperones can help them out. But this is, I think, self-assembly, and I think like that's a big problem that we're trying to model right now is protein folding in these energy landscapes.
Yeah. We can talk more about it. I have some ideas regarding chaperones and stuff.
I just remarked you probably have like 3 minutes if you're willing.
Yeah. Okay. And then we were sort of like, you know, I'm saying like stalling is sort of like widespread in these processes in biology. And what is widespread are also these, you know, backward facing error correction processes. And in the case of self-assembly, what you could have as a error correction is just that, you know, these just disassemble. So all these parts go away and you can restart from the beginning. And it's something that is known for example, when, you know, there's self-assembly of microtubules, you know, there's a sort of like disassembly process. And that's exactly what we were inspired from, called dynamic instability, which is just a sort of like disassembly of the structure. And again, so when we run these simulations of self-assembly for speed, what we see is that our structures that are the fastest are also the ones that start to disassemble, to have these dynamic instabilities, proving that, again, you know, selection for speed is giving these error correction mechanisms. So I feel that this project became a very big project that touches a lot of things, but as you know, you said before, like when you write a paper, the origin of the paper doesn't come through. And I think for me, like it's very unique the way that I started working on these, where, you know, none of these cool part was there. I was actually working on a very specific thing, and then it sort of like exploded in this project. And I feel that also what I learned a lot was to really, you know, try to, you know, sometimes there's no experimental data that can help you, so there's nothing to do. But in this case, it was really, really crucial to, you know, go and look at, you know, the data about how, you know, DNA polymerase stall and trying to understand this process, and also having the luck to have data sets that were actually measuring the two traits that we wanted and, you know, plot them and look at them. And in the meanwhile, I also turned out to be an experimentalist. So I am doing my own experiments to like measure these two traits and sort of like do evolution experiments to try to prove that if I do like evolutions of DNA polymerases for speed, I actually find more accurate ones. But yeah, I felt, you know, during my PhD, I really was lacking this experimental connection, and I feel that I'm very happy to have found it now. So yeah, I don't know. That was my talk.
Have like time for 2 quick questions or 1 serious question before we rest our brains for tomorrow.
I actually have a question. So I'm gonna take my opportunity here. But okay, so for this sort of like dynamic instability type correction, which I am assuming is sort of it doesn't necessarily matter that there was an error, but I'm gonna disassemble a portion and then reassemble it again, and then I'm gonna disassemble and reassemble, and then hopefully throughout this like long extended process, I've built like enough things that are all correct. Is that the idea there?
You're right that you disassemble even if there's no error. So the disassembly happens at a certain rate, independent of whether there's an error or not.
Like help you not have errors?
It helps you. Actually, what I didn't tell you, so yeah, we also try to abstract this even further. And I think what it helps you is that sometimes you just get lost, and you get lost so badly that there's no way you can recover. And the only way to recover is just to restart. And I think the idea is that, you know, if you are trapped in these sort of like configuration with an error for a very, very long time, it's better just to, you know, go back there and restart again. And I think this is the sort of like error correction that our work touches. Is this sort of like what means by backward facing is just a sort of like, let's restart from the beginning. And this is true when stalling effects are large, because you need to have a very large cost in making these mistakes. If the mistakes don't cost a lot of time, then there's no point in restarting from the beginning, right? Yeah. And to say these stalling effects in DNA polymerases are quite huge, because the rate of extension past a mismatched base is actually up to 10 to the 6 lower than correct base. So, you know, that is pretty impressive. And also checkpoints, yeah, I feel like it's sort of like the numbers sort of are there in biology in certain of these processes where like these mistakes are actually costing a huge amount of time.
This comes from physics or from selection, to Aditya's question?
Stalling?
Stalling for in the example of the DNA polymerase. Like,
yeah, that is, I think that is chemistry. I mean, just physics, yeah.
But you could have a genetic code which didn't have stalling or something like that. Really?
No, you cannot have a genetic code without stalling, because actually what Jack, you know, the contribution of this collaborator was that he has done experiments where he was doing DNA, non-enzymatic DNA replication, meaning that you replicate a DNA chain with no polymerase. So you just have nucleotides that attach to each other. So that is just genetic code without enzyme in a way. And that also stalls. So meaning that it's really just the way that the structure of the nucleotides that make them stall.
I could have totally different nucleotides.
Oh yeah, but the current genetic, I mean, our genetic code.
Right. But if I ask like, why is our genetic code the way it is?
Yeah, this is
the answer could be because it stalls, which then allows more complex stuff.
Yeah, this is there are people that study this, and I'm not one of them yet. Yeah, but because I think the origin, I think the nucleotide, the origin of like the nucleotides themselves is something that is more like there could be stalling, but that there are also many thousands of other things that are important for them to be that way, right? But yeah, it's true.
Okay, with that, thank you so much, Riccardo.